T1 and T2, those were samples that had cancerous activity.
Yeah.
And so A, B, C, D, E, F, G, H, I, J, those are all genes in that sample.
Okay.
Okay. And then those numbers that it's talking about, 32 through 269, those are like indicators of locations within that gene that they're looking at specifically. Does that make sense? Like you're looking at specific, and I'm pulling up here. I have it.
So the numbers are different locations, like is it like a locus sort of?
Yeah, and yes, that's exactly right. Yeah, so I've got a slide I'm pulling up here. Do you guys remember last semester? So again, intentionally bringing back stuff to review, right? Last semester, we did notes on this stuff. And this would have been section 2.6-ish, 2.7 for our notes. So if you're looking back at your old notes or looking in the textbook, you're going to find that in transcription, we have ways that we regulate that, right? So we regulate it by having like locations that the polymerase can bind onto on the gene, right? So if this is our gene here, okay, we have, you know, enhancer sequences, but really the big one here is this promoter. And if that promoter isn't working properly, then it's either going to get, let me make sure I don't want to say this incorrectly. It will likely not get transcribed. So if that promoter sequence isn't working properly, then that RNA polymerase isn't going to be able to bind on, and then that coding, that gene won't get transcribed. Why this is really important is I want you to think about when we were learning about mitosis. Remember how we learned about genes that stop cells from dividing? We learned about the P53 gene, retinoblastoma. We learned about all of these genes that stop cellular transcription that stop cellular division. And if something is wrong with that gene and the polymerase can't bind on to create the protein that stops cellular division, then you're going to get excess cell division. Right? And so I have this really cool model I'm trying to pull up, but when we're looking at this, right? So here's the image again. Well, let me pull it up on.
So is methylation something that promotes cell division or?
No, so if you remember last semester, we learned about methylation, that it's, here, I'll pull. And we'll talk about all of this too, just so that you guys can bring this all back together. So methylation, I already have this open, it's right here. Methylation is, where'd it go? So remember how we learned about DNA methylation? How if you put a bunch of methyl groups onto something, you're either turning it on or turning off the gene, right? And got this up here. Let me get this here. Oh no, not that one. Sorry guys, I thought I had this open, but I didn't. So, shoot, not that. Here it is. So let's just really quickly, I'm gonna show you guys a model of this where, loading. So last semester we learned about, oh, that sound is annoying. We learned about nucleosomes, right? And we learned how the DNA wraps itself around the nucleosome. So that's here on this picture. Remember how we have the 8 histones that bind together to make a nucleosome, and that's how the DNA wraps around it? This is kind of starting to come back a little bit. Okay. So the DNA is going to wrap itself around this nucleosome. And then if the DNA is wrapped really tightly around that nucleosome, well, guess what? It's not going to be able to be read and transcribed, right? Because it's too tightly packed. You can't get any of that transcription machinery on there. But one of the things that we can do is we can add methyl groups. So these are just little like CH3, they're just little tags, and I'll show you that. So this is a really good model of it. If we are to like take methyl groups off, notice how the DNA unwraps from the nucleosomes a little bit. It's not as tightly wound, right? And so what that does is it allows for more of that protein to be made. However, if I add methyl groups, see how tightly that DNA can wrap? And look at how much less protein is made. Does that sort of make sense? So now back to our question here. If there's too many methyl groups on here, and we can assume that this is a tumor suppressor gene, right? If there's too many methyl groups and it's not getting read, because these T1 and T2 genes look like this. And if this, instead of being green fluorescent protein, were actually a cell division repressor, like saying stop cell division, then this one isn't going to be able to tell that cell to stop dividing, right? So in our example, these cells, T1 and T2, this methyl group is likely, or these genes are highly methylated, so they're really tightly compact. They're not getting read. Whereas these N1 and N2 normal genes don't have as many methyl groups, so they're probably not getting, or they're probably getting read normally and being transcribed and translated and making that protein that causes cell division to stop. Does that sort of help?
Yeah.
Okay, so again, a lot of pieces to one question, but it's a really good practice in reviewing all this information.
Okay.
So I'll let you guys kind of put some of that into words. You can see some of your classmates have already started adding on to it. And I'm going to go check in on a couple of the other groups, okay?
Okay.
Thank you.
All right.
Thank you.
Yep. Let me. Hey, I'm going to add Allie to your group.
Okay.
She just came in, so I'm going to add her to your group. Oh, there we are. Hi, Allie. I added you to this group. I feel pretty confident that they can help catch you up. And I'm going to just in the chat box for you guys, I'm going to go ahead and send you that simulation that I was just using to show you so that you can kind of use that to help describe some things, okay?
Thank you.
Mm-hmm. Hey, folks. I'm back. How you doing?
Yeah.
So I don't know, we were trying to work on question 2, the second note box.
Mhm.
And I was looking at methylation, is that how you pronounce it?
Yeah.
So I was seeing that it controls the on and off switch of DNA.
Can you help me understand, where were you looking at to get that information? How did you get that?
I went to nature.com. I'll share the link with you.
Oh, no, yeah, that's great. I just wanted to know if it was beneficial to you on Google or if you needed to look at your notes.
Oh, I was just looking on Google. I found it. I mean, I understood, but it might not be so easy for other people. It depends, but it's the on and off switch. But if there's so many of them, it could cause it can lead to errors and cause cancer.
Mm-hmm.
So, because you can see so many in the first 2, there's more errors.
Mm-hmm.
Would that be okay? Yeah.
Yeah, let me real quick. I want to give you guys a quick breakdown of something and just show you. I'm trying to figure out why oh, there it is. In the chat, I'm going to send you guys a little, well, actually, I'll wait 1 second. I'm going to send that in just a second. I'm going to show you something, okay? So I'm going to share my screen. Okay, so what this problem is asking us, right? So this, again, I want to reinforce, this is on your quiz tomorrow, okay? So this question told us, it said that samples were taken from 2 colon cancer tumors and 2 normal colon samples. So a particular gene was implicated as a possible cause of cancer. The promoter of this gene was cloned. So that's A through J, okay? So what they're telling us here is that they are looking at the promoter sequence of this specific gene that they are isolating. So this could be like, let's say this is on, I have no idea, I'm just making this up out of my thin air, but let's say this is on chromosome 14 at locus, 1062, okay? And we're just looking at an isolated tiny little section of gene from samples of colon cancer and normal cells, okay? And so what they told us here is that section 32 to 269 is the section of DNA that codes for the promoter of that gene. Really quickly, I'm just going to remind you of what the promoter is. And these were notes you guys took last spring. So you would have taken these. It's like section 2.6, I think. I can't remember exactly. I'll have to pull it up. But you would have taken these notes over DNA regulation. Okay, it would have come in. Actually, I do want to find that real quick for you. Let me just pull that up so that you guys know, like, these are in your notes, and I want you to be able to see them in your notes as well. Yeah, so 2.6, 2.7, it would have been in this transcription section. So you would have seen it in these notes here, okay? So it was in all of these kind of boxes that I had you guys like drawing in and things, okay? So RNA modification, etc. So transcription, when we talk about transcription, and we talked about the really easy pieces to it, right? We know that there's a RNA polymerase that binds onto the DNA, the RNA reads the DNA, and it just like shoots out the mRNA transcript, right? And that's in the nucleus. Well, really what we're looking at here is, here's our gene, right? So here's a strand of DNA, okay? And then on that strand of DNA, we have our promoter. And that promoter is what attracts the RNA polymerase, right? That's kind of like where the RNA polymerase knows to bind. Now, the promoter serves as different things, right? But the promoter, if the promoter section is methylated, in other words, let's look here, right? Emma gave a great description. Notice what this DNA here in this model looks like, and I'll send you guys this model to be able to play with it. This has a bunch of little green methyl groups. Do you guys see all those little green dots on this DNA? Do you remember what these little circle things are called that this yellow DNA is wrapped around? I'm going to just show you real quick just to, again, reinforce that we did go over this last semester. So remember that whenever DNA is tightly packed around those nucleosomes, does that word hopefully ring a bell? We did a little activity. So the nucleosome has the DNA wrapped around it. And if the DNA is too tightly wrapped, like, it can't be read. There's nowhere for those transcription machinery pieces to bind on. And so it looks like this, right? So these are the nucleosomes. This is yellow stuff is the DNA that is wrapped around the nucleosomes. And then these little green things here are the methyl groups. Now, methyl groups are nothing but carbon and 3 hydrogens. Okay, it's nothing special. But those methyl groups allow, and watch what happens as I do this. So I'm untangling the DNA, and look at what's happening. We now have less methyl groups. Those little green dots go away, right? And what did you notice? Just watch these two little boxes. So here's mRNA being made, and here's the protein. So watch the boxes. So as I turn off the DNA, less protein is made, right? As I turn on the DNA or unwind it, more protein is made. Okay.
So could the methyl groups make it tighter, and then the what's it called?
Polymerase?
Yeah, the polymerase can't get on as easily because, and then the mRNA and protein can't be made.
Exactly. So
Okay.
let's change these boxes from being named mRNA and this green fluorescent protein. Okay, I'm going to actually do this real quick because I can. So instead of it being, haha, look at this. Instead of it being green fluorescent protein, right? It's now going to be a cell division repressor. So this protein, when I make this protein in this simulation, oh gosh, I'm awesome. I don't have my little pin hooked up. So this cell division repressor causes cell division to stop. These are called tumor suppressor genes, right? We learned about these last semester. P53, retinoblastoma, all of those are tumor suppressor genes that if they aren't made can lead to cancer, right? We have genes inside of us that actively keep us from getting cancer. So in this model here that I'm showing you, right now, this cell isn't going to be able to divide. We've turned off the cell division repressor. Or I'm sorry, that's wrong. Sorry, I said that backwards. Right now, this cell is dividing like crazy. Okay, because this cell division repressor protein isn't being made. But if I, whoa, whoops. If I open this up, right, if I open up this DNA, then this protein that allows for cell division to stop is being made, and that means that there's no cancer. So again, more methyl groups on this specific gene means we're having uncontrolled cell division because that cell repressor protein, the stop sign, isn't working. So when we look here, whoops. When we look back at our drawing, our diagram here, which one of these genes has more methyl groups on it? Which sample?
T1.
T1 and T2, right? They both have more methyl groups than N1 and N2.
Mm-hmm.
So what that's telling us is that those genes are not being transcribed. And so those are likely tumor suppressor genes that aren't being made that are resulting in cancer. Does that make sense?
Mm-hmm.
It's a lot. This is being recorded. Don't worry. We'll upload this and you guys can see it again. But try and put some of that into words while I go into another breakout room. Is that doable? Okay. I will see you guys in just a bit. Chat about that, and I'm going to go check on some other rooms. How do I, there we go. Hey guys.
Hello.
So I saw that you guys have been posting. I've been in other breakout rooms. Can you guys give me a little bit of insight into, oh great, so I see question 2 here from you guys. Oh, question 1. If an organism has a higher mitotic index, it is likely it has a tumor as cancerous cells divide at a much higher rate, yada, 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 yada. Does that make sense to you guys? You guys remember doing those labs where we looked at different cancer cells? Okay, good. Question 2. Whoops. Whoa. What did I just do? There we go. Oops. Okay. Question 2. Genes that are highly expressed have low levels of methyl, and genes that are expressed less have.
Wait, hold on.
Okay.
We meant high for the second one.
Gotcha. Okay, so let me show you this really quickly, okay? I want to show you guys a model here. I'm going to share my screen. So what we're looking at here is a section of DNA, okay? And do you guys remember those little circular things that the DNA is wrapped around?
Yes.
What are they called?
Nucleosomes.
Good, yeah. And just so you guys see, that's from the notes that we took last semester, right? And so DNA packaging in itself, just how DNA is packaged, is a way of controlling how much that DNA is being read, right? Because we can't have every single gene being made all the time, okay? Because we have those things. Remember, we learned about P53 last semester when we were learning about cancer. And that is a gene that controls the production of a protein that stops cell division. Okay? So P53, if that gene is being made, it makes a protein that causes cells to stop dividing. So we actively have proteins that are made to make sure we don't get cancer. We call those tumor suppressor genes, right? Also known, they can also be called oncogenes because they can be cancer causing if they're not appropriately made. And so with this model, right? So let's say that this is P53. We'll just use that one as an example, or we can call it the T1 or whatever. This thing here is using GFP because that's a really easy one. But let's just say that it's a tumor suppressor gene, right? Right now, this DNA is so tightly wound up and it has all of these little green methyl groups on it. You guys see those little green tags? Those are methyl groups that cause a cell cause DNA to be tightly wrapped up. However, if instead we remove those methyl groups, notice I removed those are gone. What happened to the amount of green fluorescent protein being made?
A lot more.
A lot more, right? So if instead of green fluorescent protein, we're saying this is P53, well, we can assume that this cell would accurately stop its division process and it would not go into cancer-causing mode, right? So if we look at our diagram here, right, we see this. Between sample T1 and T2, which of those has more methyl groups attached to it? I'm sorry, between like T1 and T2 compared to N1 and N2, which ones have more methyl groups?
T1 and T2.
T1 and T2, right? So what that tells us is that T1 and T2 are likely still condensed, okay? And that means that they likely are not being made. So that means that those are probably a tumor suppressor gene of some kind. And whatever happened in this cell to cause that extra methylation, it's resulting in the gene not being made, which is resulting in uncontrolled cell division. Does that help visualize it a little bit better?
Yes.
Okay. I'm going to go ahead and just in your chat box, I'm going to send you guys that little model that I was just showing you so that you can kind of play around with it and just get a visual of it. Do you have any other questions for it?
For the notes, I was looking through the note packet.
3.1?
Yes.
Okay.
And it seems like 3.1 is mostly about alleles and like sort of like passing down of genes rather than like methylation in tumor cells.
Yes.
Am I correct when I'm saying
You're
that?
100% correct. And if you actually look, this is our quiz for tomorrow, right? Which statement correctly describes genome and proteome? Okay. The issue with like trying to say we're only going to do a quiz over 3.1 is that like so many of the questions that the IB generates are like all encompassing. Does that make sense? So even though, yes, we're doing 3.1, the things leading up to it need to be fair game for review as well. So that's why, because this would have been really hard for you guys to come across on the quiz tomorrow. And so that's why I wanted to show it today, but it's also a great opportunity for review because we really need to review throughout the year as we build. But yeah, so you'll notice I kind of threw in like, so for the quiz tomorrow, which processes are involved in the development of cancer? Okay, that's a review question, yes, but again, like you're going to get the opportunity after this quiz is over to go back and edit your responses. And so it's more, it's nothing to like trick you guys into a bad grade. It's more for you guys to just see IB questions and practice like answering those. Is that okay? Cool. I'm going to go hop into another group. You know what? I'm just going to send you guys the quiz. And if you want to start looking through it now so that you can preview it, don't start answering the questions yet, or else it won't let you like, I'm not sure that it'll let you go back and, I'm not exactly sure what that would do, just to be honest. So just go ahead and you can review and look at the questions. Make sure that works. Yeah. So you can go through and look at the questions and just get an idea and start using your old notes as well as your new notes to make sure you're feeling comfortable moving forward, okay? Alrighty, I'm gonna go into another group and I will check in on you guys in just a minute. I should be done in just a moment here. Hi guys.
Hey, Miss Roper.
Yep.
Question for you.
Answer
I
daily
was-
double.
All right, sweet. I was looking up this methylation in DNA, and it's saying it changes the activity of a DNA segment without changing the sequence. But why is it adding this methyl group in the first place? Because does it always need to help repress gene transcription, or can you use other things like introns and stuff like that?
Oh yeah, absolutely. So if you guys remember from this last semester, we took notes over this. So this was a document here, control of eukaryotic genes. And your notes packet that you took it over was section 2.6, I believe, if you're looking back at your notes packet from last semester. But for example, there are many ways that we control gene expression, right? One of those is by DNA packaging. Oh, Jacoby just left. Okay, whatever. Bye, Jacoby. So for you two, DNA packaging, do you remember how, welcome back.
There.
Yeah,
Okay, everything just like muted out and I couldn't hear you. Sorry, go ahead.
you're good. You were gone.
Oh, okay, well, I'm back.
So yeah, Jacoby, there's a lot of ways that DNA is controlled, right? So you've got your nucleosomes. Remember how we learned about nucleosomes and we did that simulation where we like learned about the different complexes of nucleosomes?
Right.
The histones and everything. So whenever DNA is tightly bound together, it looks like this. Wait, hold on. Okay, so do you guys kind of see like on my model here, the DNA is really, that yellow band is representative of the DNA and it's really tightly bound around these blue nucleosomes.
Right.
Do you observe anything else that's on this DNA here?
The proteins.
These little green dots?
Yeah, are those proteins?
Those are methyl groups.
So what's the importance of the methyl groups?
Great. So methyl groups, watch as I unwind the DNA. Where do the methyl groups go?
Are they creating a bond, or are we looking at
They're
what's
gone.
behind
They come
it?
off.
Oh. Okay, so it's what's in the background.
Yep,
gotcha,
those are
gotcha.
what is in the background. You constantly have methyl, like methyl is just CH3. It's nothing complex or crazy. So I can show you here. Again, this is in your notes, but I'll also just share this slideshow with you guys so you can see it. So these are the notes that we took over DNA methylation, but methylation of DNA blocks transcription factors. Okay. Please don't try and get yourself too into the weeds about like how the methyl groups go on and off.
Okay.
There's a lot of different factors for why methyl groups go on and off, and it deals with signaling. Also, the environment can change whether or not methyl groups are there, but you really don't need to get into that. It's just extra knowledge. Okay. But when you attach a methyl group to DNA, okay, you're going to get like these things. It causes that DNA to tighten back up. Okay. And when it's tightened up, there's no way polymerases can actually like bind onto this DNA. There's nowhere for it to really do that and be able to read this, right? So we have to demethylate and open up the gene to turn it on to create new protein.
Okay.
Okay. So if we're looking, all of these things tie together, right? And so now you asked about splicing, like here's that splicing. Yes, that's another way to regulate it. But
Okay.
the big one we're looking at here in this question is about the promoter, right? And so the promoter region, if you're looking at my picture here, this promoter, the DNA section, you have a promoter, and that promoter is where the RNA polymerase binds on. That RNA polymerase, if it binds on, can then go on to read the coding region of the DNA. That's only if it's open. Okay, if it's not open, if it's closed because it's methylated, then you're not going to get transcription of this gene. So think about it in this way. We learned last semester when we learned about mitosis and cancer-causing molecule or cancer-causing genes, oncogenes, we learned about P53. And P53, if you recall, is a gene that causes cell division to stop. So P53.
Oh, it's a tumor protein?
Tumor suppressing protein.
So it's important to stop cancer, or is it what stimulates it?
It's what stops it.
Okay.
But if that protein can't be made appropriately, you no longer have the suppression of cell division, and cells can divide uncontrollably.
Right.
So if we look here, if we look here at my picture, right? So T1 and T2 are our tumor samples. What are N1 and N2, and N1 and N2 are our normal samples. Which one of these has more methyl groups, methylation?
Comparing the T's or the N's?
Comparing across the T's. So does group T or group N have more methyl groups?
T.
Yeah, T, right? And so,
Right.
and specifically, we can even get more specific and say T1 has even more methyl groups.
Right.
Because what we're looking at here is a strand of, we're talking about the DNA here. These are each cloned genes. So they've cloned this gene to look at across the board methylation of these genes. And this, they said in the question, section 32 through 269 specifically is a promoter sequence, right? And so if this is a methylated promoter sequence, then this isn't being, that whatever the gene region after this promoter sequence is not being made.
Oh, because they didn't get rid of the, okay, that makes sense.
Yep, so this section, whatever the gene that this promoter section is reading for, codes for, it's not being made. And so what we can assume then, just from kind of our understanding of how all of these pieces work, is that, all right, this promoter sequence must code for a gene that suppresses tumors. And since this promoter sequence isn't being made, then we have excess cell division taking place, which is causing the cancerous tumors to be made.
Okay.
Does that make some sense?
Yeah. That
Yeah,
makes
yeah.
sense,
No,
definitely.
that
little bit.
adds to what.
And does that at least can you guys see, I know that section 3.1 is about mutations and stuff, right?
Right, at the
That's what
genes
all
and
the notes are
genomes.
about. But can you
Yeah.
guys see how these things all tie together?
Right. Well, I didn't even think about the whole P53 thing and like tumor suppressants, but I mean, you're right, and that has all to do with mutations.
Exactly. So that's why we have to start tying these things back together.
Right.
Wouldn't have
Right.
quite made sense to do it in last year.